Hello, everyone. Thanks for giving us this opportunity to present our work today. This is Jiahui Yang from Sino Biological R&D department. Today, I will talk about our work in antibody discovery platforms to support functional monoclonal antibody generations. So today's talk will be divided into 4 major parts. First, we will briefly review the processes of antibody development. As immunization is the first step of the antibody development, we will talk about more about the challenges and the solutions of immunization. Then we will move to talk about the antibody discovery platforms. And for the last part, we will share our results and the data for some antibody discovery and experiments. As we all know, monoclonal antibodies is applied in many biological areas, including research and diagnosis, such as IHC, IF, flow cytometry, ELISA, and Western blot. So in addition, antibodies are also widely used in therapeutics. In 2021, 100 antibodies has been approved for therapeutic use. And there are still many more and under the preclinical and clinical development. So first, let's briefly review the processes of antibody development. Antibody discovery is a complicated and long process, starting from immunization, antibody generation for screening positive clones, and then expression and purification of in vitro recombinant antibody expression, then followed by the functional analysis. So for example, immunization consists several steps, including antigen choices and which animals we want to use for immunization and how to proceed the antibody immunization. For the antigen choices, it includes what types of antigens we want to use for immunization. If we want to use recombinant antigens, we need to figure out which expression host we want to use to express the antigens. And for the immunization, we can think about the immunization routes, timelines, and also if we want to use regular immunization or speedy immunization, which we will talk about later in the next few slides. We always believe applications determines the development strategy. So which means for some of the applications such as Western blot, IHC, those experiments are done under denaturing conditions. Therefore, the antibodies most likely recognize the linear epitope of the target. But for some other applications such as ELISA, flow cytometries, and most likely the antibody recognizing the conformational or linear epitopes. So based on the final application, we need to decide which type of antigens we will use for the immunization. There are a couple choices here. For native proteins, it's definitely one of the good choice for antigens because it contains the native structures, but due to the low yield and it's difficult to obtain some of the native samples, it has limited use. This type of antigens can recognize both linear and conformational epitopes. So therefore, we will use recombinant proteins as antigens. It has high yield, but not always the same as native proteins for the structure part. There are also, we can use peptide for the linear epitope antibody generations. It can be synthesized in vitro, so you can easily obtain a large amount of antigens for immunizations. But peptides it has low immune response, so usually you need to use a carrier protein to boost the immune response. We can also use DNA or cells. And for this type of antigens, it can produce antibodies recognizing both linear and conformational antigens. because they contain the native or close native structures. But for DNA or cells, it also has a problem of low immune response. And sometimes for the cell surfaces, they have some complicated surface proteins, which have challenges for the screening part. So here is an example when we produce an antibody targeting the specific epitopes. And this epitope is located within a high homology sequences. We want to produce an actin antibodies, which specifically recognize the smooth muscle actins. So when we search these genes in the genome, we find out there are a couple other genes shares high homology sequences with the smooth muscle actins. So we label the red amino acids here, showing that this is the only differences between these several actins. So therefore, we chose peptide as the immunogen for these antibody generations. Finally, we successfully obtain antibodies can only recognize the smooth muscle actin without any cross-reactivities with other muscle actins. As I mentioned before, immunization consists of multiple steps crucial for successful antibody development. So as the start of the whole processes, the strategies of choosing immunization procedures will determine the following steps. So we'll discuss more about this step. As I mentioned before, there are a couple antigen choices for the immunization steps: peptide, protein, DNA, and cells. For us, Sino Biological, since we have already set up several recombinant techniques for recombinant protein expressions, so we will prefer to use recombinant protein as the antigen for antibody discovery. So as I mentioned, we have HEK293 cell and CHO cells for protein expression. We also have insect cells, E. coli, and yeast expression host to support recombinant protein expressions. But there are several challenges of recombinant protein expressions, which I listed here. For the recombinant expression, sometimes we'll get low expression level. We can see that this is a protein used E. coli system to express the recombinant protein. And before and after the induction, we hardly see the target protein present in the E. coli host. We also observe aggregates after protein expressions. And some of these aggregates are insoluble proteins, such as in inclusion bodies when we use E. coli system for protein expression. Degradation is another problem we usually see during protein expression. We are seeing the target proteins located on this band, but we are also seeing some degradations after purifications. For some of the enzymes as antigens, when we use recombinant expression technologies, from batch to batch, we are seeing the variations. So how are we going to solve these problems? So here's a couple solutions to solve the recombinant antigen expression problems. So this is one example. We did vector optimization. We optimized the expression vectors to try to increase the expression level of the recombinant proteins. And here is the example. The green light shows the GFP proteins. We can see that when we use our expression vectors, we are seeing more GFP proteins expressed in different types of cell lines. We also have different generations of plasmid. We are seeing that for this Sino Biological plasmid number 2, which have better expression yield compared with other two plasmid. We usually optimize the culture conditions to improve protein yield or to solve the degradation and the aggregation problems. Here are 4 examples showing here. And this is the problem for the protein degradation. We can see that as long as the culture duration increase, we are seeing more degradations. Of course, we are also seeing more proteins expression after 30 hours incubations. But if we don't want to use the degraded antigens for immunization, we're probably gonna stop the culture expression at 26 hours. And we can also change the temperature of the expression. If we are using relatively lower temperatures, we will see fewer degradations for the targeted proteins. This is example showing the insect baculovirus expression systems by optimizing the MOI. So this is the transfection ratio of the viruses and cells. And we are seeing for this experiment MOI 3, this is not the MOI exact number, this is the experiment number 3. We are seeing more proteins expressed in insect cells. To solve the aggregation problems, we can definitely use adding additives to decrease the aggregates. So for 2 proteins, the process works for using these components. And with and without additives, we are seeing more proteins expressed in this expression condition. And we can definitely use DNA immunizations as we mentioned in the previous couple slides. And particularly for some of the transmembrane proteins, it is very challenging to obtain the purified protein from, no matter it's the natural sample or recombinant host. So sometimes we are thinking to use DNA immunizations, but the most challenging part of the DNA immunization is it always gets very low immune response. So how to solve the problems? The first part is we optimize the expression vectors, because we are thinking if we can improve the expression level of the target protein inside of the cells or the animals, we are able to get better immune response. So these are 6 different types of expression vectors. After immunized with different vectors individually, we test the serine titer targeting to the positive cells expressing the target molecule or the control cells. We are seeing that the vector number C showing the best serine titer for the DNA immunization. So in addition, we optimize the immunization procedures, including the immunization routes and also the timeline and how many times we inject the animals. And we figure out the plan 5 and plan 6 showing better titer of the serine after immunization. So therefore, we test the CD34 monoclonal antibody discovery. CD34 is a single transmembrane proteins, and we immunize the expression vector containing the mouse CD34 genes, and then screening for positive clones. And we find that the antibody we obtained are able to specifically recognize the mouse CD34 cells. And you can see the differences between the isotype control and the positive antibodies. So for the immunization strategies, we can also consider the dosage, routes, and adjuvants. So for different types, different animals, different species of the animals, we will use different dosage for injection time. And there are also multiple routes you can choose to boost the immune response. For the adjuvants, usually we use CFA. This is very common adjuvants to be used for the immunization. And for the timelines, for some of the projects, we want to speed up the whole processes of antibody discoveries. So for the regular immunization procedures, it usually takes about 2 to 3 months to obtain immunized animals. So therefore, by changing the dosage and the routes and also adjuvants, we are able to obtain similar immunization titer when we only immunize the animal within 3 to 6 weeks. So if you are seeing here, there are 12 different targets, and we are using rapid and routine immunization procedures. The green bar is for the rapid immunization method, and the gray bars represent the routine immunization procedures. We are seeing that for most of the targets, we are able to obtain similar or close syringe titer compared to the routine immunizations. Very interesting one is for the molecular, the target number 1, we are seeing better immunization response when we use rapid immunization method. Then let's move to the third part, the antibody discovery platforms. So this slide is just showing the timeline of the important events in therapeutic antibody generation, including the major antibody discovery platforms. So we are seeing that both hybridoma and phage display technologies has been used for antibody generation for therapeutic use. And for single B-cell antibody techniques, which is discovered in 1990s, we are still waiting for the antibodies for the therapeutic use, approved for the therapeutic use. So just briefly introduce the process of 3 different antibody discovery platforms. I mean, in Sino Biological, we usually use immunized animals, immunized mouse, and immunized rabbit. But for other labs, they can also use the blood samples or tissue samples from the donors, either immunized or just naive. And for the single B-cell isolation, we will first use different types of techniques to isolate single B cells. And most likely, we are isolating the antigen-specific single B cells. Then we can obtain the antibody sequences directly from the B cell cloning by RT-PCR followed by PCR. And then we can express the antibodies by recombinant techniques followed by functional analysis. For the library, the antibody library, for Sino Biological, we usually use phage to display the antibody library for screening for the positive clones. By this method, we are also able to obtain the positive antibody sequences directly after the screening. For the traditional hybridoma technology, we fuse the B cells and myeloma cells to get the hybridoma and then screen for the positive clones. This process takes much longer compared with the previous 2 techniques. So for the traditional hybridoma, just briefly go through the process. After the immunization, we'll be able to obtain the B cells from the spin, and then by cell fusion, we will screen for the positive clones, I mean the positive supernatant for 2 rounds of ELISA screening. Then we'll do the subcloning of positive ones and then screen for the positive one again. If we want to obtain purified antibodies, we can do clone expansion for in vitro cell culture productions. So based on the processes, there are multiple steps can be optimized to increase the successful rate of using hybridoma technologies. So the first one is we have been discussed previously. We can use rapid immunization procedures because the whole processes for hybridoma generation is a very long processes compared with the other 2 technologies. So we can shorten the immunization procedures. So second, for some types of applications, we can do syringe screening for positive applications and to help us to decide which animals will be used for the next step. And then to increase the cell fusion rate, usually the the cell filtering rate is very slow, and it determines the successful rate for the later. Therefore, there's a lot we can do to increase the fusion efficiency. So we use electrofusion to increase the fusion efficiency. So here are 5 examples showing here. When we use electrofusion method, we are able to get more positive clones compared with the regular PEG fusion method. Then we'll screen for the supernatant, which is by assays for the final applications. For example, if you want to use antibodies for cell-based assay, we'll definitely screen the supernatant for cell-based, which means you need to set up a high throughput method for the screening. So for us, we use in vitro cell culture for production instead of the assays for the cloning expansions. And sometimes, we will obtain the hybridoma antibody sequences for longer storage, because hybridoma is not a very stable cell lines. It may lose the antibody after several rounds of cloning or the long time storage. So this is another way to help us to save these good antibody clones for future use. So for the antibody library, the phage display, M13 is the most widely used bacteriophages of E. coli, and is also used for antibody phage display. The M13 contains 5 major code proteins, which listed here, and the most widely used for phage display is the protein, the P3. This one is used to fuse to the scFv, which is the antibody fragments for displaying the antibody sequences. Because P3, it has a relatively flexible structure, and it has ability to display large protein without losing its functions. There are major 4 types of antibody libraries: native library, immune library, synthetic library, and also semi-synthetic library. I will not go through very details for this part. So after generate this display antibody, it will go through several rounds of biopanning to screen for the positive clones. We usually just do 3 rounds of biopanning, but if we didn't obtain enough positive clones, we probably will move to more rounds of biopanning, something like 5 rounds or 4 rounds. So by incubating the display library, the phage display library, with the target molecules either coated on the plate surfaces or in solution, and then we wash by washing to remove unspecific binders, followed by elute the specific binders. And after these 3 rounds, we are able to obtain positive clones for future expression and validations. So here are a couple commonly used biopanning methods, and the immobilized biopanning is mostly used in our lab, but we also do in solution biopannings in case some of the antigens, when they coated on the plates, they will change some conformational structure, which not be able to expose enough epitopes for the further antibody screenings. But there's also other methods of biopannings which listed here. During the biopannings, sometimes we are not able to obtain some good positive clones. So we are looking for some ways to solve this problem. So here are 4 examples and solutions for biopanning processes. For some of the target proteins, we didn't get good concentrations or low amount of target proteins. For solve this problem, we'll definitely switch the coating temperature from 4 degrees to 37 degrees to help to get enough coated proteins. And we are seeing that we are able to get good screening results for the biopanning procedure. And sometimes we didn't get the good product clones. It might be due to the epitope exposures. So what we do is we will use in-solution biopanning to replace the regularly used immobilized biopanning method. If we didn't get good serine titer after immunization, we will consider to coat more target proteins for the biopanning processes, or we will try to get a new library with a relatively larger size to increase the successful rate of the further screening steps. For antibodies targeting for small molecules or peptides, we will recommend to use plates with better binding capacities. So here is the comparison data that showing that when we use the regular plates, we didn't get any good binding results, but when we switch to the plates with better binding capacities, we are able to get good OD450. So the last technology we are using in-house is the single B-cell antibody technologies. After we immunize the animals, we will isolate the single B-cells for antibody sequence cloning. So here is the flow cytometry data showing that for different types of species, we will use different procedures or protocols to isolate the single B cells, because for mouse B cells, there's are available set of antibodies to be able to select for or sorting for the single B cells. So we are using CD19, IgG1 positive, and IgM negative to select for the B cells. And we will also use labeled antigens to select for the antigen specific B cells. So for the rabbit B cells, we are only using the SSC, FSC, and then select for the IgG positive B cells, then followed by the antigen selection as a sorting to obtain the antigen specific single cells. And then we will do the antigen gene amplification after 2 rounds of PCRs, when we will clone the heavy chain and light chain sequences to the expression vector for the further recombinant expression procedures. So definitely you can use other methods which are listed here for the single cell isolations. There are a lot of references available to describe all types of single cell isolation techniques. Then we'll move to the last part of the presentation. We want to show some results when we make our antibody products in-house, and also the challenges we're facing and solutions we will use to solve the problem. And the first case is about a COVID-19 antibody development. I think I don't need to spend more time talking about what is COVID-19. So for this antibody development, we believe speed is the most important part to quickly obtain functional antibodies or specific antibodies which can be used for COVID-19 research, diagnosis, or even therapeutic use. So first, based on the sequence homology between SARS-CoV and SARS-CoV-2, here is showing that for the major molecules like the spike, they are sharing very high homology of sequences. Therefore, based on this characteristic, we rescreen the SARS antibodies or libraries for SARS-CoV-2 antibodies, because we have a large selection of SARS antibodies, and we also already construct the SARS antibody libraries. So therefore, we are able to obtain some SARS-CoV-2 antibodies, and this is showing that the antibodies are able to bind into the SARS-CoV-2 S1 protein and also the SRBD proteins. It can also recognize the ACE2 expression. HEK293 cells can infect these cells. And then here is the fluorescence showing that the virus is inside of the cells. And then we definitely want to produce some SARS-CoV-2 specific antibodies. Therefore, we have to start from the beginning. We have to first obtain these antigens and then immunize the animals and do the following steps. And the SARS-CoV-2 antigens, there are some challenging parts. For example, the spike protein is a complicated molecule, so we need to think about which expression host to be used to obtain these proteins. And the spike is highly glycosylated homotrimer. It can be cleaved by host protease, and it has open and closed conformations. So for the spike protein, we chose HEK293 cells, the insect cells, to express these particular proteins. And for the nucleocapsid proteins, the N proteins, we use E. coli system or the insect system for these particular antibodies. So we are showing here that the SARS-CoV-2 spike proteins, this is the S1 proteins, has the functional activity to bind to the ACE2, the receptor on the cells. And for the N proteins, we pay more attention about the aggregates. We are seeing that this is about 330 kilodaltons. And after 3 rounds of freeze and thaw cycles, they are still able to present it as an oligomer, stable oligomer. For the speed of the antibody development, we use the single B cells as one of the technology platform to speed up the procedures. And the whole process takes about 30 days after the immunizations. And this is for the spike antibody development. We are seeing that we are able to obtain couple clones which has neutralization functions about the spike antibodies. And for the COVID-19 antibody development, there's another problem for that is the specificity. Because as I mentioned before, there's a high homologies between the SARS-CoV-2 and the previous SARS. But there's also have other coronaviruses, which is the common coronaviruses listed here. They also share some sequence homologies. But when we want to develop the SARS-CoV-2 specific antibodies, this is the problems. And so what we do is we try to enlarge the antibody libraries. And second, we use multiple platforms to support the antibody development in order to obtain enough antibody clones. So here is showing the anti-spike and anti-N antibodies. We successfully obtained the antibodies only recognized the SARS-CoV-2 viruses. Even now, the SARS, the original SARS and the other 5 coronaviruses. And the same thing for the anti-N antibodies. As we also know that the SARS-CoV-2 has a relatively high mutation rate. This is because of lack of the mismatch repair mechanisms. So these mutations or the variants may affect biological functions, and most importantly, it causes concerns of prevention because the original available antibodies may not be able to detect the newly observed variants, or may not have the function to neutralize the newly observed variants. So here is the couple examples of the variants showing here. The spike proteins and the N proteins are the 2 major molecules for the therapeutic use and the diagnosis. So to solve this problem, first what we do is we make a large collection of variant antigens to support of new antibody discoveries, because we want to find antibody which is able to recognize all types of variants. In addition, we do the animal immunization. We optimize of the procedures. We use conserved fragments or we will immunize multiple antigens to try to obtain antibodies which covering or has the ability to recognize all types of variants. The same thing, we use multiple platforms to support or get more potential antibody clones, which also can recognize diverse epitopes. This is an example for anti-N antibody development. These anti-N antibodies are able to, several anti-N antibodies are able to recognizing the variants in the ELISA assays. And for the anti-spike antibodies, we focus on the neutralization functional, and it is able to neutralizing different variants of the spike. And the last challenges for the COVID-19 antibody development is the scale up, because this is gonna not have work a lot. Like for example, for the diagnosis area, they need a large amount of antibodies for producing the kits. So we did some process optimizations and get the stability assay showing that even after storing at 37 degrees, we are able to obtain the same SEC purity as the original ones. And for the last two cases, we want to focus on the neutralization antibody generations. And the first case is we are using the hybridoma technology to support the neutralization antibody generations. And we definitely immunize more animals. Usually we just immunize 5 mice, and this time we immunize 10 animals. And then we are setting up a cell-based neutralization assay to screening for the clones. So after the immunization, top 2 animals for cell fusions, we are able to get 44 top ELISA positive clones, and to conform with the flow cytometry, we obtain 8 parental clones conform with this assay. We did 2 rounds of subcloning, and finally we obtained 6 flow cytometry positive subclones like for the antibody productions, and then validated by the cell-based neutralization assays. And this is the TIGIT antibody discovery. The TIGIT is a crucial molecule for therapeutic antibody generations. This time we use the antigen-specific B cell sorting. We obtain the single B cells and directly do the cloning for the antibody expression, followed by the validations. Within 26 days, we obtain multiple clones or multiple antibody sequences, which showing similar function as 2 positive controls. So just to summarize today's presentations, and for all 3 antibody generation platforms, they both have their characteristics. For the hybridoma, this is the traditional method for antibody generations, and it can produce native chain pairings and also full-length antibodies. But we need to do a lot of work to increase the fusion efficiency of the cell fusion, and also the development time for hybridoma is much longer compared with the phage display and single B cells. For both phage display and single B cell, these 2 techniques didn't rely on immunizations. They can obtain a large antibody diversities. But for phage display, this is not full-length antibodies. Usually it's scFv and Fab, and this heavy chain and light chain are not native pairs. And for single B cells, the problem for this is depending on the single cell sorting technologies. And also it's a big cost for doing the RT-PCR and the sequencing. So therefore, there are multiple antibody development platforms available for antibody generations, and it will help us to get all types of antibodies. And it depends on which application you want to use. So you can choose either of them for antibody generations. So the last part, just briefly introduce what Sino Biological is doing. As probably you already know from the previous slides, we are a company producing reagents, and we are also using our technology platforms to support CRO services. We have more than 6,000 proteins available, and also more than 13,000 antibody available. All these products are developed using our in-house recombinant expression system and antibody generation platforms. We are supporting CRO services from protein expression, antibody discovery, and we can also do biological assays and large-scale productions. And thanks for your time. And I would like to answer any questions. And here's also the contact. If you have any further questions, you can contact our local office. And thanks.